Thank you for having me here. Clearly a different institution. There's a different slide color. So
that's how we can tell.
There's some logos too, maybe. So my lab studies tumor immunology. I've come from a basic and translational research background, and we're really interested in doing this in pancreatic cancer, and for good reason. There are literally no really good effective treatments for pancreatic cancer. This is one of the best ones we've seen in years. And what you can see from this graph is a new chemotherapeutic called FOLFIRINOX does extend survival for 4 to 5 months, but the end result for the majority of these patients is the same. This is considered one of the better studies in 5 to 10 years. So with this in mind, we asked what is the clinical and challenge to treating these patients, and why are these tumors so resistant to effective therapies that might work in breast or prostate cancer? And this is one of the answers. These pancreatic tumors are embedded in a unique, an organ-specific tumor microenvironment. This is illustrated when I start to highlight the tumor cells here in green. And what you can see is the tumor cells are less than 1/3 of this tumor. This is a tissue sample from a human pancreatic ductal adenocarcinoma, but it's sort of representative of many of the tumors that we get in the clinic. So these tumor cells are surrounded by the stromal microenvironment. So the tumor microenvironment, the cells within there, we call them the stroma, that actually can support tumor growth and progression and even blunt the efficacy of cytotoxic therapies. My lab is particularly interested in the immune cells that are present within these tumors. And as you can see here by this brown staining, this is staining for one subset of immune cells called macrophages. They're highly present in this microenvironment that supports tumor growth and progression. And this is just another example showing you these subset of immune cells called tumor-associated macrophages that are in red, intimately associated with the tumor cells in green in this human tissue, and cross-talking with them and supporting their growth. In addition to this barrier to treatment, like other tumors, these tumors have a subset of tumor cells that are resistant intrinsically to common cytotoxic therapies. They're often referred to as cancer stem cells or tumor-initiating cells. They have elite properties in terms of their ability to metastasize and to not respond, to survive common insults such as chemotherapy or radiation therapy, shown illustratively here. But in the laboratory, we can find these cells in human tumors and isolate them. We can find them in mice and isolate them. They're shown here by the green staining in this human histological section. And they too are embedded in this unique tumor stroma composed of immune cells and other types of cells. So in the laboratory, we've been trying to study the interaction between your immune system and this subset of cancer cells, the cancer stem cells. And we focus on one immunologic subset of cells called tumor-associated macrophage. It's one of several immunologic cells present in these tumors, but it is the most abundant one present in the majority of these pancreatic tumors. And we found 3 things in the laboratory when I came to get funding from you guys. First, that these macrophages are actually predictive of poor, when in large numbers, predictive of poor clinical outcomes in pancreatic cancer. Second, that they often correlate with increased numbers of cancer stem cells, which are more resistant to cytotoxic therapies, in human patient samples. And third, that they can secrete cytokines that in vitro and in vivo in mouse models actually expand the number of these resistant cancer stem cells in tumor models and murine models. So with that in mind, we wanted to take a pharmacologic approach of targeting this subset of stromal cells present in these pancreatic tumors and see if we can improve the outcome of chemotherapy. We partnered with industry to do this. We took advantage, we knew some of the biology of this subset of immune cells, and we chose the CSF1 receptor as our target. By partnering with industry, we could take advantage of clinically developed inhibitors that were originally developed actually for arthritis, but for the most part have been stopped in terms of their clinical development for that, and we can repurpose this drug for cancer. What we find is when we treat mouse models with a CSF1 receptor antagonist, we target these macrophages, we reduce the number of the macrophages present in these tumors, but we vastly alter the immunologic response that the stroma has to these tumors, instead of supporting tumor growth, we're actually beginning to reject these tumors. This alone is actually not effective enough to restrain tumor development in mice very much, but in combination with chemotherapy, this is a very effective strategy in mice. We see increased cell death in the tumors. We see decreased numbers of tumor cancer stem cells, and we see decreased tumor metastasis. This is shown here schematically on the top, but this is real data from our published paper this year, showing you a decreased amount of total tumor burden here in the far bar and decreased metastasis. So this was actually, I'm a translational basic scientist. This was an exciting enough result that in combination with their own studies, our partnering and collaborating industry partner directly launched a phase 1 clinical trial on this year. Trial numbers there, they're using this exact approach, targeting CSF1 by targeting the stromal cells to reduce the support for the tumor, and then giving a common chemotherapeutic now, Gemabraxane. Hopefully they'll be enrolling patients by the end of the year. So in conclusions, thank you. This type of research foundation funding is critical, particularly for young scientists such as myself and the other 4 people you've heard today, to really allow us to go after big questions and innovative research. And it's already led to, sorry, the formatting's a little bit off. It's already paid off. It's already led to 3 publications this year based on some of the research that is funded by this foundation: cancer research, clinical cancer research, immunotherapy. And then we've translated those research papers and findings into 3, let's say, NIH-style grants that we submitted within the last few months. So it's been very effective. The last thing I would say is that we often think about targeting the tumor itself. There may be an additional strategy we can utilize, particularly in pancreatic cancer, in targeting the tumor microenvironment to release the support for these tumor cells such that the great agents that my colleagues have talked about can really get after it and really do damage on these tumor cells. And we think that's a reasonable approach, and we're interested in studying it.